Okay, so here we're going to cover regulation of hormones. And we're going to talk about insulin and glucagon again. And keep in mind that it's important to keep glucose levels at about 80 to 100 mg per deciliter, which is approximately 5 millimolar of glucose circulating in the blood. And then we're also going to talk about how insulin is made, as well as what its structure is and the importance of pancreas. So first off, let's talk about what happens after a high carbohydrate meal. So one thing to keep in mind when you're comparing different compounds, you always have to pay attention to the X and Y axis to make sure they are comparable. So first we have the X axis, which is giving us time, and we can see that this time is in minutes, and so that is going to be the same in each of these graphs. What is different are going to be the actual values on the y-axis as well as the units. Now the units we're not really going to pay attention to, but we are going to pay attention to the values on the y-axis. So the first thing to note is that with respect to our insulin, this never is going to be at zero because you have to maintain blood glucose levels. So after a high carbohydrate meal, you are going to get a spike of insulin that will then come down to the levels that need to be maintained in the blood. Now, if we look at glucagon, we also see that it does not go down to zero. Glucagon never is completely out of the blood circulation. It's always going to be there, ready to respond when insulin levels go down. And you can see that you might have initially a spike of glucagon that quickly comes down, but again, never back to zero. Now, after a high carbohydrate meal, you have insulin levels that are really non-measurable in the blood. And these levels will peak after a high carbohydrate meal, and then they will come down. Now, after a high protein meal, this is going to change somewhat. Again, we have our same x-axis. We have our different y-axes, and they're kind of spread out in this way, in this particular figure. And one of the things to point out initially is that glucose stays pretty flat when you have a high protein meal, when you do not have much carbohydrate in that meal. Now, if it's a mixed meal, you might get a spike of glucose, which will then lead to a larger spike of insulin. And you can see our insulin levels are much lower as well. This only goes up to 20, whereas when a high carbohydrate meal, it went up to 120. So insulin does get a small bump, but it's not a spike as you would see after a high carbohydrate meal. After a high protein meal, again, our glucagon levels are going to spike, and our nitrogen levels are going to increase. Now, the nitrogen levels will have to be balanced, so nitrogen in has to equal nitrogen out, and that will be balanced by excretion of nitrogen. Now, in our metabolic homeostasis, remember, metabolic homeostasis, main thing is to maintain blood glucose levels for the function of our red blood cells and some brain cells. So insulin is going to promote storage and utilization of fuels for growth. So remember, hormone levels are important. So insulin is going to be anabolic for fuel. And then we're going to have glucagon is going to be catabolic for fuel. And why I say fuel is because a cell needs to do what a cell needs to do. If a cell has to make proteins in order to survive, even if it's in the fasting stage when glucagon is signaling, the cells are still going to make proteins that they need to survive. So when we're talking about insulin and glucagon, we're really talking about where is the source of energy coming for that cell to maintain its function during either the fed state or the fasting state. And so during the fed state, when you have insulin, the fuel for the cell is coming from the actual foods that you eat. And for glucagon, the fuel for the cell is coming from breaking down of storage of fuel sources. Now, there also is going to be nerve input. So we're going to have the central nervous system that has signals also. And the signals can be various. It can be from stress to sleep. So there are many signals that the central nervous system is also going to input. You can have input directly to muscle from your acetylcholine receptors, and this will promote movement, and that can also trigger different mobilizations of fuel. So we have epinephrine and our other hormones that are going to be released. So we have epi, and these are going to be released. And this is going to be via things like stress. It can be sleep rhythms. It can be hypoglycemia. And then your brain, your red blood cells, your kidney medullary cells, your muscles are all going to depend on glycolysis. And so about 190 grams of glucose is required per day. And your brain is going to take about 150 grams of this. And so what that means is that the brain during a normal cycle of fed fasting is going to take up a lot of the glucose that you are going to take into your body. Now keep in mind the red blood cells are dependent on glucose as well. So that's when you go through periods of fasting, your brain switches to picking up ketone bodies and sparing it for the red blood cells because the brain would take up too much during a prolonged fasting stage. Now again, homeostasis, glucose homeostasis is going to be critical. We do not want to be hyperglycemic or hypoglycemic, so maintaining blood glucose levels is essential. Now keep in mind that only insulin and glucagon are going to be released in direct response to changing levels of fuel. So this means that in your pancreas, your beta cells and alpha cells of your pancreas are going to be very sensitive to the fuels that are surrounding or bathing those cells. Now cortisol, epinephrine, norepinephrine are going to be mediated by those neural signals. And so those are going to be sort of at need basis. So insulin is really going to drive fuel stores. And then your glucagon stress hormones are really going to drive fuel utilization, breaking down of fuels. And then, of course, in our diet, we're going to talk about the take-up of carbohydrates, fats, and proteins. And so there are only certain things that your body can take up, and those are, of course, going to feed into this fed fasting cycle. Now let's just talk a little bit about what the major organs with respect to our homeostasis are doing. And so I want to spend some time on the liver, the muscle, and adipose tissue. So here we have our liver, and over here we have insulin present. So in the presence of insulin, there are a few things that your liver is doing. Your liver is going to take in glucose. So glucose is going to go in, and glucose is going to become either glycogen for storage, or it's going to be packaged into fatty acids, and this is going to be packaged into our very low density lipoproteins. Now, our liver is also able to pick up amino acids, so during a fed state you will also be eating proteins, fats, and your amino acids can be taken up. The protein can be used by the liver, or it can be broken down into glucose and then stored again would be glycogen. Typically, it doesn't get broken down in the fed state, so this pathway is usually blocked. but it can use that protein for building something, for example. Now, in the liver, when you have glucagon present, or epinephrine, and no insulin, which is important because, as we know, insulin can override glucagon and epinephrine, this is where your liver cells do not pick up glucose. So glucose is out, which is shown here. And basically, your glucose is going to be made in your liver cells. So you're going to tap into your glycogen stores and break them down to glucose. And this is going to go to release of the glucose. Your liver does not use glucose for fuel. Your liver is only going to use fatty acids. So fatty acids are in. And remember here, our fatty acids were out. Oops. So that's different. So some key differences, let's put some numbers on here. We have number 1, our glucose in, and our number 1 down here is our glucose is out. Our number 2 is our glycogen is stored. Number 2 is our glycogen is going to be broken down. Number 3 is our fatty acids are made. Over here, our fatty acids are used for fuel. They are not made. They are being taken up and used by the liver for fuel. And then number 4, we use our proteins, and here our proteins are broken down into glucose. And so keep in mind that when insulin is present, that means that there's plenty of glucose, so the liver does not have to maintain blood glucose levels. And in the fasting stage, insulin is gone, so now the liver thinks I have to maintain blood glucose level. So the major role of the liver in the fed and fasting stage is to maintain blood glucose level. Okay, so now let's go to our adipose tissue, our fat cells. And so again, we have the same story. We have our fed state where we have insulin presence. So we have insulin. And in our fed state, we're going to have enough glucose, so your fat cells are now going to pick up glucose, and they can use it for fuel. Your adipose tissue, is going to pick up those very low density lipoprotein particles that your liver made. So this is coming from the liver. And it's going to break it down into fatty acids. And the fatty acids can go to make your stored fat, which is your triacylglycerols. And of course, your adipose tissue will use your glucose as fuel as well. And then in the fasting stage, where you have your glucagon and epinephrine, your adipose tissue is going to break down your fats and release the free fatty acids, and it's also going to release glycerol. Now the glycerol is going to go to the liver, and this is going to be used for making glucose. Your fatty acids are going to go to the liver and muscle, and this is going to be used for fuel. Now, so your liver is pretty straightforward. Your liver's goal is to mobilize fuel. And this is in the fasting stage, or store fuel. And this is in the fed stage. All right, so some differences here is that 1, your glucose is going to be taken up. Your very low density lipoproteins are going to be taken up. and one over here is no glucose taken in. Now remember, adipose tissue has the GLUT4 receptors. So this is insulin sensitive. So without insulin, your adipose tissue cannot take in glucose. And then the second difference is we're going to be storing fats, and here we're going to be breaking down fats. And so that's really what the adipose tissue does. It's going to take in things during the fed state to store, and it's going to release fatty acids during the fasting stage to mobilize fuel for the rest of the body. And then our third organ that's very important for fuel homeostasis is our muscle. And so with respect to our muscle cells, we're going to have, again, in the presence of insulin, you're going to have your muscle cells pick up glucose. This is very important because when you take in a high glucose meal, your muscle cells are going to be really key to getting that glucose out of your blood so that you don't become hyperglycemic. Also, they can take up amino acids. They can basically take up whatever is around them. Now they're going to use their amino acids and they're going to make proteins. They're going to use the glucose and they're going to store it, or they're going to make ATP and make fuel. Now in the fasting stage where you have glucagon, or more glucagon, it has epinephrine signaling, but so we'll cross out epinephrine with respect to this, but with glucagon signaling, you're going to have no effect. Muscles do not have any, so they have no glucagon receptors. And this is important because it's going to keep glycogen stores for need. So remember, glucagon is going to mobilize fuels, and if you're not using your muscles, there's no reason for your muscle cells to tap into their glycogen stores. They want to keep that for when you actually need them. Now, what is the muscle cell going to take up? It's going to take up your fatty acids, or it's going to take up your ketone bodies. And it's going to use this for fuel, and it has no glucose use. And this is because, again, it has that GLUT4 receptor, which is insulin sensitive. And so here, again, pretty straightforward. In the fed state, you are going to take up amino acids and glucose. And in the fed state, or the fasting state, over here, you're going to take up fatty acids and ketone bodies. All right, so insulin has some counter-regulatory hormones. So we're going to have our cortisol, and your cortisol provides for changing over long term. And so this is going to stimulate amino acid mobilization. And so you're basically going to break down muscles for this. It's going to stimulate gluconeogenesis. And it's going to stimulate fatty acid release. Oops. Now your epinephrine, norepinephrine are going to mobilize fuels also. And these are going to have some sort of acute state, so acute stress. And so you're going to get your glucose being released from glycogen stores, and fatty acids from your adipose tissue. Now, your secretion with respect to these stress hormones during low blood glucose is going to be one of those signals that are going to tap into your central nervous system. So you're going to have your hypothalamus, which is a major regulatory center for sending out signals to the body. And you're going from the pituitary, have your adrenocorticotropic hormone released. And this hormone is going to stimulate the release of cortisol, and cortisol is going to be released from the adrenal cortex. And then you're going to get epinephrine from the adrena medulla. And norepinephrine is going to come from nerves. There is also, with respect to low glucose level, you'll have a minor role in the release of glucagon. And of course, your glucagon will be coming from your alpha cells of your pancreas. All right, so now let's spend a little bit of time talking about your pancreas. So the pancreas is going to be a cluster of small glands. And they're going to have the islets of Langerhans scattered among the cells of the exocrine pancreas. Okay, so here's our islet of Langerhans cells within the exocrine tissue. Now, it is exocrine because it makes and secretes digestive enzymes. And this is going to be secreted into the small intestine. And these are going to be the Asner cells and the duct cells, and also connective tissue, vesicles, and nerves. And so this is going to make up about 95% of the mass. Now it's going to also be an endocrine organ, and it's going to make and secrete insulin, glucagon. It's also going to secrete somatostatin. and also something called the pancreatic polypeptide. Now you're going to have cells. We're going to have our alpha cells, our beta cells. We also have gamma cells, or what are called F cells. So you have your alpha cells. That's of course for glucagon. You have your beta cells for your insulin. And you have your gamma or F cells. And this is going to be about 1 to 2% of the pancreatic mass. Okay, so the function of our polypeptide that is made by your endocrine portion of your pancreas is to regulate the endocrine and exocrine secretion of the pancreas. And so secretion is going to increase after a protein meal, fasting, exercise, and hypoglycemia. So this guy Okay, so now let's talk just a little bit about our hormones. And we're going to discuss just very briefly insulin and glucagon. So insulin and glucagon are polypeptide hormones. These are going to be synthesized as a prohormone. So you have a proinsulin here, for example. And then you're going to have a pro-glucagon here, for example. And you can see that they're very long and big, and they're going to get modified to be their active forms. Now, glucagon is going to be cleaved into mature glucagon within storage vesicles. And this occurs within and of course, this is the alpha cells of the pancreas. You're going to go from that pro-glucagon to glucagon. And pro-glucagon also contains other peptides, and these are important during fasting, and these are glucagon-like protein 1 and glucagon-like protein 2, as well as glycantin. Here it is, glycant. Oops. So GLP-1 equals glucagon like peptide. And these are primarily going to be made in your actual digestive tract. So these other hormones that are made within the pro-glucagon can be made in different cells. It doesn't have to be in the alpha cells. Glucagon itself is going to be processed in the alpha cells of the pancreas. Now, proinsulin is going to be cleaved into insulin and the C-peptide. So we have our insulin here, and then we have our C-peptide that's going to get cleaved off. Now, your insulin is going to be in vesicles, and they're going to precipitate with zinc. And you're going to also require calcium to mobilize the vesicles in which glucose is in. Now, insulin is going to be secreted when you have blood glucose levels increase, of course. And so insulin secretion depends on glucose, the presence of zinc, as well as calcium. All right, so we know a lot about the release of insulin because of diabetics. So let's talk about how insulin is released. So there are what are called proximal events and distal events in glucose sensing in your pancreatic beta cells. Now keep in mind that the beta cell is sensitive to what's occurring in the actual pancreas, not what's occurring in the body as a total. And so that's important to note. It's actually what we call bathing the pancreas. And so after a high glucose meal, you are going to take up glucose. And then this glucose is going to enter the metabolism of the beta cell. And you're going to generate, of course, ATP. And so glycolysis, entry into mitochondria metabolism, these are all referred to as the proximal events. And so this is sort of what every cell does when it picks up glucose. It's going to break glucose down into the glycolysis pathway, then go into the mitochondria and do oxidative phosphorylation and generate ATP. Now the distal events are what actually lead to insulin secretion. And a few things have to happen for distal events to occur. One is you're going to have to change what's going on at the membrane, so you're going to have to excite the membrane. Two is you're going to have some form of calcium signaling. Remember, I told you in the prior slide that insulin release is dependent on both zinc and then calcium for signaling for its actual release. And then you're going to have your granulocyte recruitment and exocytosis of your actual insulin and release of the insulin. And so keep in mind, insulin's going to be released by beta cells of the pancreas. So let's go through each of these. So in the first step, you're going to have those proximal events in which you actually take up glucose, and this is through that GLUT2 receptor. Remember, GLUT2 receptor is constitutive, it's always there, but it has a very low affinity for glucose, so it only will pick up glucose if there's high levels of glucose. And so the glucose is going to go through its normal process, glycolysis, oxidative phosphorylation, etc., and you're going to accumulate ATP, energy. Now, after you accumulate this energy, you are then going to target the ATP-dependent potassium channel. Okay, now exit of potassium through the open potassium channels hyperpolarizes the membrane, right? So point 1. Hyperpolarizes the membrane. Okay, point 2. Closing potassium channel depolarizes the membrane. Okay, and so what you want with respect to the membrane to release insulin is you want depolarization. Okay, and so what basically that means is that you are going to inhibit the potassium channel. Okay, and so ATP, an increase of ATP is going to block So let's put it here. Increase ATP shuts down the channel. And then in 3, you have a depolarization. This is step 3. And this is going to lead to an open calcium channel. And you get an influx of calcium. And so now you have your insulin, which is in a vesicle with our zinc. And our insulin with zinc will then be recruited to the surface of the membrane, and it's going to exocytose and release insulin. Okay, so let's just look a little bit at that potassium channel because that's very important. Many drugs to treat type 2 diabetics, for example, will target this potassium channel. So the potassium channel is referred to as an octamer. It has 8 parts. And it's going to have 4 what are called identical Cure 6.2 units, and that's going to form the core around the potassium channel. So you have these Cure 6.2s. And then you're going to have 4 identical SUR1 subunits. So these are the SUR1 that are sort of encasing the Cure 6.2. So mutations in these channel proteins can lead to defects in the membrane potential. So for example, mutations in Cure 6.2 are found in neonatal diabetes mellitus, and it results in a constant open potassium channel. And so this is going to be leading to an inability to release insulin. Now, typically when ATP levels rise, you're going to have a shutting of the potassium channel. And so one thing that is going to be able to treat your diabetics are sulfonylurea drugs. Sulfonylurea drugs are important because they're going to bind to that SUR1, and this is going to allow those channels to close. So it's sort of forcing the channels to close. And the sulfonylurea drugs are going to be what are going to be used to treat type 2 diabetics. All right, so let's just talk a little bit about regulators of insulin. So again, insulin is going to be released if there is glucose present. So this is a major effect. And then minor regulators are going to be amino acids. You'll get a slight spike of insulin. You're going to have some neural signals that help to coordinate insulin release. Some amino acids are going to be able to do this. You have gastric inhibitory polypeptide, or GIP, a gut hormone after food intake that can help prepare your pancreas for the release of insulin. So there are going to be things that can help during, besides just glucose. But it is a major regulator is, of course, going to be glucose. Now, with respect to glucagon release, this, again, remember, glucagon never goes down to 0. So glucagon is going to be rapidly mobilized. In the liver and kidneys, especially when your insulin levels go down. And then remember, the liver has to maintain blood glucose. And your kidney cortex is going to maintain glucose for the medulla. So your kidney cortex can also do gluconeogenesis during the fasting stage. And so both of those are going to have to rapidly be turned on to making glucose in order to maintain glucose levels in the blood or glucose levels in the kidney. Major regulators are going to be, of course, negative by insulin and glucose, positive by amino acids. And then your minor regulators are going to be those nerve inputs.